hi, a very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So welcome to my new beginning. So this is part 4, Mendelian Genetics. So we have been talking about the unit genetics and evolution recently. So we are continuing with where we stopped yesterday. So that is on Monday. So we are having YouTube classes on alternate days, Monday, Wednesday and Friday. So in the previous lecture, so I gave you around, or I guess 10 to 15 questions as homework to solve, so that you get strong in your basics and the logics, and you also improve your time, because when I have a genetics quiz, so then it will be very helpful. So that is the sole reason I gave homework questions. So today let's see how to solve those questions. I hope you guys were able to find my PPT, able to find these questions and solve the questions. So now let's see how you have solved, and I will solve along with you, so that you know where you have made a mistake, where logically you were wrong, and how you have to approach. Right? So then I'll teach you a small bit about mutation, then we'll go forward. On Friday we have a Menti in human physiology as promised. So Friday we'll play a Menti quiz at 4. Then Saturday I have term 1 paper discussion at 4 p.m. live. So Saturday term 1 bio paper is there, so I will do the live discussion at 4 p.m. And then Monday we'll continue with genetic disorders, where I will discuss in detail about pedigree analysis, how to approach the questions of color blindness, sex-linked inheritance, etc. Alright, so shall we move forward? Yes, Mendelian Genetics Part 4. So we are going to solve the homework questions that I gave on Monday. So let's see how are the questions and how are we going to solve them. Right, so we are going to solve the homework questions here. So question 1, 3 types of gametes formed by the genotype. So I have a dihybrid genotype heterozygous condition, and what are the gametes that will be formed with the help of these genotype? So let's see. Yeah, here we go. So we have RrYy. So how do you find the gamete? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So there are 4 gametes that are formed. So now, which among the following is matching exactly? So capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So this is absolutely matching. So option A is the answer. So if you guys have solved the question, so option 1 would have been your answer. So I have the genotype of one of the parents here. So this parent looks like a heterozygous condition. So it is like your F1 offspring, right? So this F1 offspring, so what are the different types of gametes it can have, right? So let's move forward. So question number 2. In law of independent assortment, how many factors are involved for a dihybrid cross? So law of independent assortment is based on a dihybrid cross. So dihybrid, di is 2. So there are 2 characters that are involved. So since they're asking how many factors, it is 2. If they are, yeah, so it is 2 characters. So you have your seed shape and your seed color. So question 3, so the question seems pretty big, so it occupies almost the full slide. But let's see, it's pretty simple. In Mendel's experiment with garden pea, round seed shape, right? So this is one character was dominant over wrinkled seeds. Fine. And yellow color was dominant over green color. So RrYy is dominant over small rYy. So this is round shaped and yellow color. So this is wrinkled and green color. Right? So this is the question that's given. So what are the expected phenotypes in the F1 generation of the cross? So the cross is given. So in the F1 generation, what will be the phenotype? So let's do. So I have the parents. So the parents are this. Let me rewrite it again. So these are my parents. So now how do I choose the gametes? So for this R, so 1 R is my gamete, this Y, 1 Y is my gamete, right? So with these R, 1 R is my gamete, with these Y, 1 Y is my gamete. So the cross between this would be like this. So only round seeds with yellow cotyledon, only wrinkled seeds with yellow cotyledon. So here you get only 1 member. So this is only a round seeds with yellow cotyledon, so that is only your dominant character. If you're smart enough, looking at the cross itself, you can write down the answer. You don't have to do so much of work. So they're asking you, in a dihybrid cross, how will the F1 offspring be? Of course, the F1 offspring will be dominant, so the answer is option A, right? Fine. So here we come to question 4. In a cross between pure bleeding pea plants having yellow round and green wrinkled seeds, find out the total seeds having yellow color in the F2 generation. So yellow color is dominant here, right? So in the dihybrid cross, so this talks about 2 characters, so it's dihybrid cross. So the phenotypic ratio is 9:3:3:1. So this is round yellow, right? So this will be what? So if we are talking about the first character, so it will be round yellow, so this will be the other vice versa, right? So if you talk, so this will be your round, so this can be round in green, so this will be wrinkled yellow and this can be wrinkled green. So this is completely recessive type, right? So how many of the seeds will have yellow color? So 9 plus 3, that is 12. So 12 out of 16 seeds will be yellow in color. So here the right answer is option A. So question 5, so in a cross between pure breeding plants, so we have the same plants, what will be the seeds ratio of seeds having yellow and green seed color? So we have, so what will be the ratio? So whenever the ratio is asked, which means we're talking about the F2 generation, so having yellow and green seed color. So we have, so let's see. So in the dihybrid, in the F2 generation, we have 9:3:3:1. So this is yellow and green seed color. So here 12 will be green and this 4 is wrinkled, right? Sorry, this will be yellow and this will be green. 12:4 is nothing but 3:1. So the right answer is 3:1. So this is how you saw. So directly 12:4 is not in the answer, so you might get confused. So these are yellow and these are green, right? So that is how I've taken 9 plus 3 is 12, 3 plus 1 is 4, right? So question 6, if the dwarf pea plant was treated with gibberellic acid, it grew as tall as the pure tall pea plant, okay? If this treated plant is crossed with a pure tall plant, then a phenotypic ratio is likely to be. So it was initially a dwarf plant. If I give gibberellin, only the phenotype changes, genotype does not change. So it is nothing but a test cross, it is nothing but a back cross, right? So here the ratio will be, so 50% dwarf and 50% tall. So here the right answer is option 2. So question number 7, in sweet pea plants, the presence of dominant C and P genes is essential for the development of purple color. The ratio of plants producing flowers of different colors in the progeny of the cross will be, okay? So you need capital C and capital P to produce purple color, right? So here the parents are given. So here the parent will be, sorry, here the gamete will be this, but here the gamete is only 2, right? So you can do the cross and you find out. So here it will be capital C, capital P, small p. So definitely this is recessive, this will not have. So you have 1 purple plant, 1, 2, this is also purple, 3, 4. So you have 4 purple. Okay, so I guess we have 5 purple if I am not wrong. Yeah, so 1, this is recessive, this is, so this is not purple, so this is not purple, so this is small c. So we have 1 capital C, 1 capital P, so this is purple. So here both are small p, so this is not purple. So here I will do the purple in the purple color, so it's easy for you to understand. So this is 1 purple color flower, this is 1 purple color, so you have 2, and here we have a 1 purple color, right? So we have almost 3 purple, right? And 1, 2, 3, 4, 5 and 5 white color flowers. So this is option D. So this is how you solve questions of this type. But if you start practicing, yes, you can do it at a faster rate. So question 8, Mendel crossed a pure white flowered recessive pea plant with a dominant pure red color plant. The first generation of hybrids from the cross show. So he is taking a pure pea plant. So pea plant does not show incomplete dominance, right? So he took a white colored recessive pea plant. So and red color was dominant. So in the F1 generation, all will be red flowered plants. So this is according to your, so we are asking in the first generation. So here everything will be your F1 tall plants. So that is here the dominant one is red plants. So from a cross given below, the following genotype ratio that will be obtained in the F1 generation. So this has been repeated n number of times, be it NEET, be it your term 1 exams. This is very important. So you have to take these two parents. So to find out the gametes is pretty easy. So here the gamete can be capital A, capital B or small a, capital B. So here you have only one type of gamete. So the offsprings can be this or this. So which one correctly matches? This. So this is your answer. Yes ma'am, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Right, so let's move forward. So we are here in question 10. So I guess I have solved question 9. Yeah. So we move to question number 10 now. A dihybrid for a qualitative trait is crossed with homozygous recessive individual of its type. The phenotypic ratio is. Ma'am, the question might look complex. No, it is not. Let's read it again. A dihybrid for a qualitative trait is crossed with a homozygous recessive individual of its type. So I have one dihybrid parent. Say for example, this is my dihybrid parent. Okay, it is crossed with its homozygous recessive type. So this is your test cross. So your dihybrid test cross ratio is 1:1:1:1. So instead of directly pointing out a dihybrid test cross ratio, they have made it like this. So this is option C. Right? So question 11, the percentage of heterozygous individuals obtained in the F2 generation from the selfing of plants with the genotype RR would be. So selfing of plants, so if you self these two, percentage of heterozygous that you will get will be 75. Sorry, it will be 50. So this is 25, 25, these two are 50. So heterozygous is capital R, small r. So here it is option B. So we move on to question 12. In a dihybrid cross, 80 plants are obtained. Okay, so in a normal dihybrid cross, we get 16. So here 80 plants are obtained. The ratio of homozygous and heterozygous shall be. So in your dihybrid cross, we have 9:3:3:1. Right? Yes. So out of this, this is my homozygous. So 10 is my homozygous and 6 is my heterozygous. Right? So 10/16 into 80 gives me the number of homozygous and 6/16 into 60 gives me the number of heterozygous. So if you do it, you will get it. So this is 8 2s are, this is 10s are. So 100/250. So this is 2s are, 10s are. So this is 60/2, this is 30. So this is 50:30. So which can be 30:50 because first they are asking you homozygous condition. The ratio of homozygous and heterozygous shall be, okay. So your heterozygous condition will be, so this is, okay, so here we are not supposed to consider the phenotypic ratio, we are supposed to consider the genotypic ratio. So it will go vice versa. So your homotypic ratio will be 30 and your heterozygotic ratio will be 50 here. Alright, yes. So question 13, the F2 ratio 9:3:4 is explained on the basis of epistatic gene. So I haven't taught you what is epistatic gene. So epistasis, so I have two genes working on two different genes. I have a gene A and I have a gene B, and one gene on this this side masks the phenotypic expression of the other, that is epistasis. So one is overpowering the other, so that is epistasis. Alright, yes. So let's move forward to question 14. A self-fertilizing trihybrid plant forms. So if it's a monohybrid plant, the total offspring is 4. So if it is a dihybrid and if there are 4 gametes, you form 16, right? Sorry. So let me write it like this. If it's a monohybrid cross in the F2 generation, you get how many offsprings? 4. Right? So in a dihybrid cross in the F2 generation, you get 18. Sorry, you get 16. Hi Ranjan Maheshwari, hello. So thank you so much for joining my lecture today. So I am discussing questions from genetics. So this you get 16 offsprings. So in a trihybrid cross, F3 would be, so what is this? 4 power 1, 4 power 2. So now 4 power 3. So what is 4 power 3? 16. So you have 8 different gametes and 16 different zygotes, right? So hi Prince, a very happy good evening. So thank you for joining my lecture today. So please do like, share and subscribe. So Prince, you told me you have pinged me in Telegram, but when I checked, I saw no message. Maybe you have texted someone else, so please get back to me so that I will get back to you, right? So if you are in the NEET 𝐒𝐴𝐴𝐴 point channel, because I never got your message, so I scrolled right after the class on Monday, but I didn't get your message. So maybe you have texted someone else in the name of Krishna. So if you are in the 𝐒𝐴𝐴𝐴 point NEET channel or in that group, so find Krishna. So I am one of the admins. So text me and I will get back to you because I don't have any of your message. So I searched Monday afternoon also, evening also I did, and now also before class I did check, but I could not find your message. Alright? Yeah. So here we go to question number 15. The phenomenon in which an allele of one gene suppresses the activity of other gene is known as epistasis. So this is option C. Yeah, so with this we are done with all the homework questions. So I have solved all the homework questions for you. So I guess inheritance of one gene, inheritance of 2 gene, linkage, sex determination, all that is clear. So we are going to talk about one small portion today, that is mutation. So what is mutation? So we have already discussed. So sudden or discontinuous variation in the genotype and phenotype of an organism due to the change in the base pair of DNA is your mutation. Along with recombination, mutation also leads to evolution. So your coronavirus is very fastly mutating. So that is why you had alpha, beta, delta, then you have recently the Omicron strain, right? So mutation is actually good, but it can take up, mutually is actually needed for evolution and variation, but mutation can either be good or either be bad, right? So there are 3 types of mutation depending on the cause. The first one is gene mutation, the second one is chromosomal aberration, and the third one is genomatic mutation. So let's see what is gene mutation. Alteration of DNA due to the change in the nucleotide sequence. So one nucleotide sequence is replaced by the other, or it's deleted or it's substituted. So that is your gene mutation. So when more than one nucleotide pair is changed, or when you have more than 1 point mutation, it is called as gross mutation. Alright. So gene mutation occurs by the following method. No, that is not me. That is not me. So you have my name as Krishna. Okay. Right. So the frame shift mutation. So when I have a frame shift mutation, so for example, when I write red hat or the red hat, so now it makes sense to you, right? But if I write, so if I delete the D and write the, so does it make sense? No, it changes the way we read, right? So this is my frame shift mutation. So suppose if I have another word here. So this also changes my reading frame because your codons can be read in a triplet form only. So it can read only 3 words at a time. So substitution, when you replace one by another, it is substitution. So substitution can be of two types, transition and transversion. So your nucleotides are of two types, one is purines and one is pyrimidines. So purines are A, adenine and guanine, pyrimidines are cytosine and thymine. So when an adenine is replaced by a guanine, that is when a purine is replaced by a purine, it is transition. When a purine is replaced by a pyrimidine, it is transversion, right? So chromosomal aberration, loss or gain of a segment of DNA results in the alteration of chromosome. If you see, this is my normal chromosome, but one segment is lost, this is deletion. So here you see, so here the terminal portion is deleted, so this is terminal deletion, and here the intermediate portion is deleted, this is intercalary deletion. So these are different ways that mutation can occur. So always it's very clear, right? So small things make big difference. So one small change in a single base pair in your DNA can change the complete phenotype, right? Fine. So what are genomatic mutations? A change in the chromosome number where we have visible effects on the phenotype. So sometimes when you have a mutation, it has a direct effect on the phenotype. That is, it is expressed in the person, it is expressed in the body. So like in case of a person born with a genetic disorder, it is directly expressed in the phenotype. You can consistently or distinctly see the person is affected, right? So there are some mutations which will not be expressed in the phenotype as well. So far they are saying Omicron virus is not very lethal, is not very severe, it is not getting expressed phenotypically. But your Delta, your Beta versions were pretty serious, right, of your coronavirus. So much in the same way. Alright. So yeah, so change in the chromosome number which is visible in the which has a visible effect on the phenotype. So there are two types of genomatic mutations. One is aneuploidy, one is euploidy. So what is aneuploidy? That is due to the non-disjunction of two chromosomes of a homologous pair. So in an aneuploidy, either you have extra chromosome or you have one chromosome less. So the chromosomes do not separate properly. So that is your aneuploidy, right? So when you have one extra chromosome or one less chromosome. Trisomy, tri is 3, so you have 3 copies of a particular chromosome. So ideally we are supposed to have only 2 copies, but when you have 3 copies of a chromosome, that is trisomy. So you can also have monosomy in case of your Turner syndrome, where the person or the female has 1 chromosome less, right? So these are genomatic mutations. So what is euploidy? So euploidy is when you have more set of chromosomes. So now we have one complete set, so one from the father, one from the mother. But in plants, they can have 3-4 sets of chromosomes, that is euploidy. So plants are literally polyploids. A change in the number of chromosomes, the multiple number of chromosomes that they have, this is due to the variation in one or more diploid sets of chromosomes. So this is aneuploidy. So with this we are done with mutation. So what is remaining in Mendelian genetics is genetic disorders and pedigree analysis, which I will begin on Monday. So Friday we have a Menti quiz in human physiology. So we are going to have I guess 35 questions, 5 questions from each chapter, 25 seconds for each question. So please do join me for my Menti at 4 p.m. on Friday. The topic is human physiology. So I have recently finished human physiology, so you can go through my lectures. We have already played a small Menti before, so this is going to be a big Menti. So please join me for my Menti on Friday at 4 p.m. and Saturday at 4 p.m. we are having term 1 paper discussion. So this is all I have for today, because I thought I will solve questions and that itself is saturating, right? Okay, so if you guys genuinely like my way of teaching, so please do like, share and subscribe, and Prince Kumar, so see if you can find me and ping me for your doubts. So thank you so much for watching my lecture today, and this is me signing off until tomorrow, until Friday at 4 p.m. So thank you and bye.